Hi, my name's Sam,
Hi, my name is Alyse,
and we're here to talk about DNA structure and base pairing.
DNA is made of a nitrogenous base, a deoxyribose, and a phosphate group. The nitrogenous bases are in between, and they're held together by hydrogen bonds, and then the backbone is made of the deoxyribose and the phosphate groups. Rosalind Franklin used X-ray diffraction to produce a picture of DNA. And from that photo, Watson and Crick were able to determine that DNA was a double helix. The width of the helix and the spacing of the nitrogenous bases was also deduced from the picture. Watson and Crick's model predicts that when a double helix replicates, each of the two daughter molecules will have one old strand from the parental molecule and one new strand. This is called the semi-conservative model. The two strands are held together by the hydrogen bonds between the nitrogenous bases. So right here, between the bases, there are hydrogen bonds that hold them together. It's also shown in this picture. You can see between the bases, there are hydrogen bonds. The strands are anti-parallel and run 5 to 3 and 3 to 5. And what that means is that if we had this model laid out like this, this would be 5 end, the 5' end, this would be the 3' end, and this would be the 3' end on the other strand, and the 5' end. So they run opposite directions. The sugar phosphate backbones are on the outside of the helix, so that's right here on the outside. The sugar in the backbone of it is obviously deoxyribose, and they are held together by phosphodiester bonds, which is the linkage between the 3-carbon atom of one sugar molecule and the 5-carbon sugar of another molecule. These phosphodiester bonds form the backbone of DNA. And this is a picture of those phosphodiester bonds that hold the backbone together.
Now we're going to talk about base pairing. First off, DNA has 4 nucleic acids: adenine, thymine, guanine, and cytosine. Go canoeing anytime is a helpful way to remember these base pairing rules. The A, adenine, always bonds with the T, thymine, and the C, cytosine, always bonds with the G, guanine. Cytosine and thymine are pyrimidines that bond with purines, adenine and guanine. Pyrimidines only can bond with purines because there's not enough space for 2 purines to fit within the helix and too much space for the pyrimidines to get close enough to form a hydrogen bond. But when the pyrimidines and purines bond with each other, it's a perfect fit. Chargaff has 2 base pairing rules. First is the composition of nitrogenous bases varies within the species. Where you can see on this, humans and chickens, the percentages vary between the 2 of them. But within a species, the number of adenine and thymine bases are equal, and the number of guanine and cytosine bases are equal. So within the humans, the percentage of adenine and thymine are closer, and then the percentage of guanine and cytosine are the same. But between the humans and the chickens, the percentages vary between them. As a visual aid, you can see that thymine forms a hydrogen bond with adenine, and guanine forms a hydrogen bond with cytosine, and it goes all the way down the double helix. And I hope that's helpful.